In this last part of the lecture, we will talk about assembly evaluation and error correction. Obviously, one wants to be able to compare different assemblies, for example, to choose between several alternatives produced by several different algorithms. The most common metric and the most important one is so-called N50 metric. It is defined as the largest contact length L, such that using contacts of length L or greater accounts for at least 50% of the bases in the assembly. It is equivalent to a weighted median of all contact length. It is calculated as follows. We order the contacts by decreasing size, and then we start picking the largest contacts, and we look how much of the total assembly length these contacts cover. And as soon as we cover 50% of total assembly length, we register the length of the last contact that we took, and that's going to be our N50 value. For example, suppose we have a 1 megabase genome, which is covered by the assembly that we have, and we have the contacts of the following length. We have one very large contact that covers a quarter of the genome, and then contacts of decreasing size. Then we start picking the contacts and adding the number of the base pairs that they cover. And we need to go down to size 22 kilobase pairs to cover over 500 kilobases. When comparing different assemblies with N50, we assume that either the assembly covers the whole genome or all assemblies have the same length. Other metrics that can be used are, for example, number of contacts. Obviously, it is better to deal with assemblies with fewer larger contacts. So the size of the largest contact is also a metric that can be used. The total length of the assembly, the total number of bases in it. This is NG50 is an important variation of N50. It is computed equivalently, but here, instead of covering 50% of the assembly length, we aim to cover 50% of the genome length. For this, we either need to know the genome size from, for example, a reference genome, or we estimate it in a way. There are software tools that can be used to compute all these metrics for a given assembly. Now, what one can say about scaffolding. Why would we bother to do scaffolding? This is because it increases the continuity of the assembly, which can be measured by N50. Here we have two examples of human genomes from an Asian individual and an African individual assembled using SOAP de novo assembler. And here we see how the N50 value increases when we are using paired end reads. The more paired end reads we use, the better. And also what one can see here is that the longer the insert size in the pair end read is, the better. Different assembly algorithms and assembler tools have been compared by independent groups, for example, in the Gauge community experiment. You can see that none of the assembly algorithms is the best. Here we have metrics for different assemblers here for the human chromosome 14. And this part of the table is related to the metrics on contacts, and this part of the table is related to metrics on scaffolds. What we have here is the sheer number of contacts or scaffolds respectively, then the N50 value in kilobases, the number of erroneous contacts or scaffolds, that is, when pieces from different parts of the chromosomes were erroneously joined into one contact. And finally, here we have the corrected N50 value, that is the N50 that would be reached if we corrected all these errors in the contacts and we would just break erroneous contacts into pieces. And you see that if you have a lot of errors, the N50 value decreases. So the continuity of your assembly decreases after error correction. What one can see here from this table is that different assemblers excel in different values. For example, some of them are very good in producing large contacts, but they introduce a lot of errors. Others are not so good in producing the contacts, but then they are very good in scaffolding. And finally, the fewer errors you produce, the smaller are your contigs. So there is no single way to choose the best assembler, and this has to be assessed every time given the particular biological task at hand. Finally, we turn to the topic of correcting sequencing errors. We've seen how these errors get corrected at the consensus step of the OLC procedure for assembly, but it would be even nicer if we could correct them beforehand. It would make our graphs smaller and would introduce fewer spurious edges into them. Suppose that we know the genomic location for every read, and every position in our genome is covered by multiple reads. Then we can use consensus from the multiple alignment of these reads to correct the errors. For example, here we have these many reads coming from this genome, and for each read there is a unique location in the genome where it comes from. Consider this last part of the genome. We have 3 A's in a row, and in these 3 reads, 1, 2, and 3, we have 3 A's in a row at the end. So there is a CT before the A's in this read, and there is a T here, so these reads align well. But in this read, there is an additional C between the CT and the A's. So given this multiple alignment, we can assume that this C here is an insertion error. And analogously, we can correct all other errors in all reads. But this works only if we know from which part of the genome our reads come. In practice, errors in the reads are not distributed uniformly. Illumina sequencers tend to create mismatches and more of them towards the end of the read. Other technologies, such as PEG-Bio, rather introduce insertions and deletions, especially in stretches of the same nucleotides. One consideration that is helpful for error correction is that if you produce many reads covering a particular position in the genome, and the errors are rare and are distributed more or less randomly or following some random model, then in most of the reads, your position will be correct. So for a reasonable K, the frequency of erroneous K-mers, the ones that have sequencing error covering this position, will be low. And this indeed is a case. This is a typical distribution of frequencies of K-mers with certain coverage. So here we have a coverage for a K-mer, and here we have the density, which is normalized frequency. What you see here is a nice and normal distribution around the coverage of your sequencing experiment. And here you see a very heavy tail towards 0, which means that we have some reads with K-mers that occur only once or twice. So these K-mers most probably are those erroneous K-mers, and we want to correct these errors. We do error correction using so-called spectral alignment. The set of K-mers with sufficient support provided to them by the number of reads in which they are found can be called the spectrum of a sequence. Our goal is to find such a sequence from which all possible K-mers will lie in this spectrum. This procedure can be called spectral alignment. In practice, that means that we substitute all K-mers with coverage below a certain cutoff with their Hamming neighbors that have coverage above the cutoff. This is an outline of such an algorithm. As input, we receive the reads R and a coverage cutoff C. And as output, we aim to produce the corrected set of reads. On the first step, we build a hash table from R which stores all K-mer count pairs. And then for each read, we look at all K-mers in this read, and if the count for this K-mer, so the value in the hash table, is lower than the cutoff, then we substitute it with its best Hamming neighbor. And we will see how we can choose this best Hamming neighbor in the next few slides. So how do we choose the K-mer with which we are going to correct the erroneous K-mers? Consider this set of 5 reads, of which one contains a sequencing error. This will be read 5, and the sequencing error will produce 3 erroneous k-mers, for k equal to 3 in this case. We build this hash table from all the reads, and then we just register the count of all k-mers in all possible reads. And for these 3 erroneous reads, we have the count of 1. So these are our candidates that we will try to substitute with something better. For an erroneous k-mer, we generate all possible Hamming neighbors, and then we look up the counts of these Hamming neighbors in the hash table. In this case, in fact, only this Hamming neighbor is in the hash table, but this is a very small hash table. In practice, you will have more Hamming neighbors in the hash table, so we will choose the one with the highest count, which gives us the outline of the procedure for finding best Hamming neighbor. At input, we have a k-mer v, coverage cutoff c, and hash table h. At the output, we aim to find the highest count Hamming neighbor of v that has count above c. So we store these values in 2 variables, the best sequence and the best count, and initialize them with v and cutoff -1. So if we don't find such a best Hamming neighbor, we're just going to return v, and as a signal that we didn't find it, we will return this value below the cutoff. Then for each index in the k-mer and each letter A from the alphabet sigma in which we are working, we construct a Hamming neighbor v prime by replacing the letter I in v by A. And then if the hash value for v prime is larger than best count, then we update the best sequence and best count. And that in the end will produce us the best Hamming neighbor. In real-world implementation, you can improve this algorithm in several ways. For example, you can make the choice of the best k-mer consistent along the read. The erroneous base appears in k adjacent k-mers, so you might just pick the substituting k-mer so that these k adjacent k-mers will be consistent with each other. In the previous implementation, we assumed independence of adjacent k-mers, which of course is not correct. We can also use quality values for the reads that we can obtain from the sequencer. The instruments that are used for sequencing can assign a value to each read of how good it is, the instrument thinks. And we can prefer the k-mers that come from the reads with high quality. We can also disregard repetitive k-mers, for example, k-mers containing stretches of the same nucleotide or tandem repeats, as well as low entropy k-mers and k-mers that have unreasonable coverage, because these k-mers will most probably mess up the assembly anyway. So in summary, the spectrum alignment-based error correction is used in many assembly algorithms that are used also in common practice. The idea is that we replace erroneous k-mers with most similar k-mers with high frequency. Since repeats artificially increase the count for k-mers, as we have discussed before, this leads to false positive corrections, so it is meaningful to disregard these k-mers. The advantage of this approach is that it is simple to implement and easy to parallelize, because we are considering k-mers independently from each other. The drawbacks is that it works for Hamming errors, so it is only good for correcting errors introduced by Illumina sequencers. Another disadvantage is that the parameters of the correction algorithm, the cutoffs and the k-mer size, are arbitrary, and there is often no automated procedure to adjust them. So, to summarize the lecture today, we have introduced different genome assembly paradigms very briefly, and then we concentrated on the OLC assemblies, which are finding Hamiltonian paths in overlap graphs. Then we touched upon how you can do scaffolding to deal with repeats and segments of low coverage, and we have seen that it is very non-trivial. And then we briefly touched upon error correction of reads before assembly using spectral alignment of reads. In the next lecture, we will consider in more detail the second assembly paradigm that uses de Bruijn graphs. Thank you, and see you next week.